Good evening. We are delighted to have here today Dora Angelaki. You can hear by the name that Dora was born, not very far from this country. And Dora tells me that this is her first visit to Israel. So we are proud to have you for the first visit in Israel. Dora comes from the United States. One of our alumni is working with Dora now, and we just discussed how well he's doing there. Adam Zaydeh, who was a student of Sagai Bergman. She is now a faculty member here.
I hope so.
Adam is towards the end of his postdoctoral training with Dora, and we regularly meet in Greece to swim in the sea and talk about science. And today, Dora will tell us about optimal integration of sensory evidence, building blocks, and canonical computation. And Dora comes, I didn't say yet, she's a chair of the Department of Neuroscience, Baylor College of Medicine, Houston, Texas.
Yeah.
Welcome.
So thank you, Elona, for the invitation to come here. So I was visiting the Weizmann Institute, and
They cannot hear.
Oh, sorry.
We can.
I thought my voice is strong enough. Maybe not for Israelis.
I don't know. Now.
Everybody everywhere except Greece tells me, shh. So it's really a pleasure to be here. I debated whether to talk, so I really have two main areas of interest. One is the cerebellum and the other is the cortical areas. And I debated which one to present today, but so I chose this because I was afraid I gave the same talk at the Weizmann, so I thought, I will be too stressed preparing another talk. But there is another reason, and I will tell you the reason shortly. So the work I'm going to present today was done, most of it, in collaboration with Greg De Angelis. We were both colleagues at WashU when we started this work. And let me give you a little bit of an introduction. So when I started, so many of us study different sensory systems, and the smart ones study one of the 5 canonical sensory systems, and those have a discrete representation in the cortex. Oh, thank you. A discrete representation in the cortex. They have the primary sensory areas. When I started my graduate work, very soon I fell in love with a different system, a difficult one. It's the vestibular system, which measures accelerations as we move in the world. And unlike the other sensory systems, it's very difficult to study because there isn't a pure vestibular sensation, a pure vestibular perception, and there is no single area in the cortex that one can actually find vestibular signals on their own. Instead, vestibular signals exist along with other sensory signals and motor signals. So even as early as the first synapse in the brain, the vestibular information is combined with efferent copy signals and visual signals, and most importantly, somatosensory signals. So in recent years, because I'm interested in the vestibular system, of course, the last 10 years, I've become very interested in understanding what are the principles underlying how we combine different information together. So today I'm going to talk about a particular behavior, a perception, which we call heading perception, which is like knowing which way we move in the world. It is something we do every day. We do it without even thinking. And of course, important cues come from the visual system. The pattern of retinal motion, which is known as optic flow, provides a very strong cue to the direction of our motion. But of course, vestibular cues, particularly from the otolith organs, which measure linear accelerations. So in our lab, we have built virtual reality systems for monkeys and humans, and more recently also mice. So everything I'm going to tell you today will involve 3 different stimulus conditions. When the platform moves without any visual stimulation, I'm going to refer to it as the vestibular stimulus condition. When the platform is stationary and there is an optic flow stimulus generated, I will call that the visual condition. And then, of course, we are going to put the two together, and that will be the combined stimulus condition. So we train our monkeys to perform a simple one-interval, two alternative force choice task. So we move the animal along one of multiple directions that are symmetrically placed around straight forward. During the movement, for these experiments, I'm going to tell you the animal makes no eye movements. At the end of the movement, two targets appear, and the task of the animal is to look to one or the other target, depending on whether he thought he moved to the right or to the left of straight ahead. So that's the task, and this is an outline of what I'm going to tell you today. There will be two parts of my talk. The first is really trying to understand how different sensory signals, in this case, optic flow and vestibular signals combined. My lab works very closely with computational neuroscientists because I want to understand the brain. I'm an engineer by training, so I want to understand the brain in terms of the canonical computations it performs. So the first part of the talk will be that, and it's mostly, but not completely, unpublished data. Now, for the second part of my talk, I want to refer to a few other questions which have to do with what are the neural correlates of perception, and more importantly about neuronal variability. So all of these questions have emerged from this work because this is a perception that we are studying. Now, I want to tell you before I move on, specifically today, there are two important components in understanding how signals should combine together. The one component, which is what I'm going to talk today, is multisensory integration, and I will tell you what I mean by that in a second. But there is another very, very important part, which is multisensory calibration. How do we go about living our lives when one sensory system, because of disease or because of alterations, because of glasses, can change throughout our life? How can these systems actually be coordinated again? And this is something that Adam Zaidel, one of your students, has been done in my lab. So I've decided not to talk about this today because I want him to talk. I hope you will invite him to come give a job talk and he can talk about this. Okay. Oh, she's coming? Okay, perfect. Okay, so I'm specifically going to talk about multisensory integration, and this in a way is the background of what he will talk about. So our question for the first part of the talk is, how do sensory information combine and why? So you're going to hear a sound, and you try to see what the person said. Oops, wrong. It is very difficult, right? Because there is background noise. Did anybody hear the word? Not obvious. Now? So you see how much faster you are and more accurate in understanding what this person said when you now have another sensory signal present. So it's this I'm sorry?
What about without audio?
You will, I do not know. You will still guess, yeah. Can still guess, but you're not going to be sure. So this is what I'm going to address today. How can sensory signals interact to improve the signal-to-noise ratio or the precision of perception? And I'm going to define a few things for you. First, I would like to define a concept which is known as the sensory likelihood, which you can think of it as being the observer's belief about the stimulus based on sensory signals. And you can think of it as, in a very simplistic thinking, you can think of it as a Gaussian distribution, which has a mean value, which is the best estimate, but which also has a variance associated with it, and it's that sigma, the sigma square actually, that I'm going to define as uncertainty, and then the reliability, which is what I'm going to refer a lot throughout my talk, is just the inverse of that uncertainty. So I want you to remember these two definitions, and I want you to appreciate that a signal which has a narrow likelihood and a small sigma is characterized by a high reliability or small uncertainty, and in contrast, a broad sigma, a large sigma, has a low reliability, high uncertainty. So with this concept now, let me tell you how we vary in these experiments that I described to you, this heading discrimination task. We vary the uncertainty of the visual stimulus, which is very easy to do by manipulating the visual motion coherence. That's 100% stimulus. This is 50% coherence. This is 20% coherence, which basically we add noise to the visual stimulus. And for all the experiments I'm going to tell you, we actually did not vary the motion of the platform because that's much more complicated. So when we started these experiments, which is about like 8 years ago, we were lucky because there has been quite a bit of work both in computational neuroscience, but also in human psychophysics, which had thought about the question, well, what's the best way to combine the sensory signals? So under certain assumptions, which are not that unreasonable, the best way to combine the sensory signals is by a linear weighted sum, which is shown here. This is one Q, this is the other Q, but those weights are not randomly determined, but are actually directly proportional to the reliability or inversely proportional to the uncertainty as measured by the threshold, by the sigma of that particular stimulus. And of course, the two weights sum to 1. So the reason that this is the optimal solution is that because one can show that this type of combination results in the largest improvement in the sigma. Remember, small sigma is good. Improvement in the sigma means sigma gets smaller, and this is the equation. And when the sigma, the two sigmas are the same, this is where one gets the largest improvement, which is a factor of a square root of 2. So what does that mean in practice? I will show you in a second, but let me remind you what I already said previously, that we were lucky that this theory had been developed and had been shown in many sensory, multisensory examples. However, until we did these experiments, we did not know how the brain does that. And more importantly, we did not really know how the brain achieves this computationally. In particular, how does the brain carry information about this sigma, about the reliability? So this is data from the first animal we trained to do this task. This is now 8 years ago or so. So I'm going to show you all the behavior in the format of a psychometric function, where the x-axis is the direction of the stimulus, 0 is straightforward, positive is rightward directions, negative means leftward directions, and this is the proportion rightward choices that the subject makes. So we train the animal to perform this task in darkness without a visual cue, and typically thresholds are around 2 to 4 degrees. And then we vary the visual motion coherence, as I showed to you previously, such that we get this approximately the same threshold, the same sigma. These are cumulative Gaussian fits to the data, so we can measure the threshold, the sigma. And then under these conditions, when we present the two stimuli together, we see a steepening of the psychometric function, and when we fit the cumulative Gaussian, we get a smaller threshold. So this is an example of a behavior where now we can quantify how much more precise we can be when we can combine sensory signals, like the audiovisual example that I showed to you previously. Of course, one can do this at different visual motion coherences. So what I showed you was a situation where we typically the visual cue is much more reliable than the vestibular. And so what I showed you basically is true when the visual motion coherence is actually pretty low. So this is a situation where the visual and the vestibular thresholds, single cue thresholds, are about the same. The orange shows the prediction based on this equation, which is lower than the single cue thresholds. And then the cyan is what we measured experimentally, and this is the case that I showed you previously. However, I want also to point out that now when we present the visual stimulus and the combined stimulus with a higher coherence, then as you can see, the visual cue is so much better than the vestibular, such that adding the vestibular to the visual does not do much better. The prediction and the actual are very close to the visual. So this is what this equation basically tells us. So this is what happens with the thresholds, but remember that what accompanies this equation is also the weighted linear sum. So we can, in fact, also look at the perceptual weights, the behavioral weights that the brain puts on each of the visual and the vestibular cues, and this is the predictions based on this equation. Remember, we measure the two sigmas, which are just the two single cue thresholds, and this orange is just this prediction. And we can also measure it experimentally by putting the two cues in conflict. And this is what the green shows the experimental data. And I want to highlight two things. The first thing is that the animal, like previously shown in humans, they actually, trial by trial, we interleave these different motion coherences. The animal does not know what comes next. So even when it happens trial by trial, you can see that the behavior changes such that subjects, monkeys in this case, but the same is true for humans, change the weight, the perceptual weight they put on each cue. So that's one observation. The second observation that I want to highlight is that, as you can see, it's close to the prediction, but it's not exactly the same. At low reliabilities, the vestibular signal is overweighted from what it will be predicted by this equation. And I want you to remember this because I will come back to this in a second. So this is the behavior. So far, we knew pretty much all of this from human psychophysics. So our interest was really to understand how the brain does that. And there are two areas in the cortex that one finds multisensory visual vestibular signals. And these two areas are the two areas I'm going to talk to you today. One is called the medial superior temporal area. It's in the extrastriatal visual cortex. The other is the ventral intraparietal area, which is in the intraparietal sulcus. And I'm going to show this to you also in the coronal section. This is the MRI of one of our animals. This is the superior temporal sulcus. This is area MT, and this is area MST, and this yellow area is VIP. And for those of you who know the lateral intraparietal area, that's this green. So this is the areas we are going to focus today. And the reason we went there is because, as I mentioned previously, neurons there are multisensory. They respond to both optic flow and inertial motion. So we record from these neurons while the animal performs the heading discrimination task in each of the stimulus conditions. This was the vestibular. There was nothing here. And then interleaved, we also have the visual stimulus condition, which again, we record the activity on each trial. We average data from many trials. We generate tuning curves. And of course, we do this for the vestibular. I will quickly show you the data. So these are really tuning curves recorded while the animal performs the task in the three different stimulus conditions. So the first question we want to know is whether the cells become more sensitive like the animal's behavior. And to do this, we use a very well-established analysis based on signal detection theory, which basically takes these responses and converts them into a different format. The x-axis is the same, but now the y-axis is not firing rate any longer, but instead it's the proportion rightward choices that an ideal observer will make in choosing left versus right based on the sensory signals, based on the responses of this particular cell. So we assume a mirror cell, yeah. We fit then the cumulative functions exactly the same, such that we can measure the neuronal thresholds now, and as you can see, this is an example of a cell which actually became more sensitive when the two signals were presented together. As you can see from the cyan, what that means is that the cell can discriminate smaller differences in the direction of self-motion. So this is summary data. This is the behavior. This is the neurons. This work actually was published several years ago, done by a postdoc in the lab then, who is now in Shanghai. And the important point here to highlight is that similar to the behavior, we found that neurons in area MST have exactly the same property. As you can see, the neuronal threshold now is significantly lower than both single Q neuronal thresholds, and the same as the prediction. Now, more recently, another postdoc in the lab who has also gone back to Shanghai, she did the same recordings in area VIP, and I just want to show you that pretty much we got the same result. So throughout my talk, I'm going to be implying that activity in one or maybe both of these areas actually is responsible for generating the perception. So the question is whether, I'm going to address this question from multiple perspectives, and the first one, of course, we need to, the first question we need to answer is what happens if we take the population activity of all the neurons that we recorded from to see whether we can actually predict the behavior. And for this initial part of the analysis, we are going to use a maximum likelihood decoder, which of course has been used multiple times, and it's good to give this talk in front of Chaim. So this is the behavior. I've already shown to you this previously. This is the neuronal thresholds, and this is the perceptual vestibular weights. By the way, this is work done by a graduate student in the lab, Chris Fetch, who is finishing his postdoc now. So now I'm going to show you what happens if we take the cells he recorded from, we use this maximum likelihood decoder and see whether we can predict the behavior. And this is the answer. And needless to say, when Chris first showed this data to us, we didn't believe him. We thought he was just playing tricks on us, because as you can see, it's amazing. So here we took the activity of the neurons in the vestibular stimulus condition, the visual stimulus conditions, and the combined. We computed predicted thresholds based on the neurons' single Q thresholds, and as you can see, what we found has exactly the same properties as what we found in the animal's behavior. And this is true not just for the thresholds, but also for the weights. In this case, these are neuronal decoder weights. And what was more peculiar is that we could even reproduce this peculiar departure from optimality. So of course, when we saw this, we really wanted to understand what is this. I mean, these are only 50 cells that we used to make this decoding. What's special about these properties that allows the decoder to do this?
These are the cells that recorded simultaneously?
I'm sorry?
These cells were recorded simultaneously? Or?
No, I will come back to this. So these are neurons from area MST. They were not recorded simultaneously, but we have measured noise correlations. That's the second part of my talk. For now, I can just tell you that the noise correlations are not that strong, and if you take them into account, you get similar results. So it's not as important for this part of the talk. So what I'm going to show you next is maybe 3 or 4 slides trying to explain to you why this happens. So we are going to follow this conceptual diagram. So we have these neurons, the unisensory neurons from somewhere combining into these multisensory areas we are recording from, and somehow this activity is decoded to generate perception. So what I have shown you so far is that at the level of the animal's behavior, one can see this linear weighted sum being obeyed. And I have also showed you that when we take the population activity of all the cells we recorded from, then we can actually predict something which is very similar to what we saw in the behavior. The next question we are going to address is whether it actually has two parts. The first part is that can we even write, if we take the firing rate, the tuning curves of these cells, do they follow this linear weighted sum also at the level of individual neural firing rates? And then, of course, the second question is, can we say anything about those neural weights if assuming this equation fits them and assuming we measure those weights, how close are they, the measured weights, to the optimal weights which will allow us to do optimal Curie weighting? So
you're doing optimal linear readout here for the population?
We're using maximum likelihood readout. But for this part that I'm going to show you next, forget about the readout. We are just going to do the analysis on a cell-by-cell level.
But R1 and R2 and R3 are rates now?
Yes.
Not likelihood?
Yes. They are rates. They are firing rate weights.
So you are trying to model the responses as linear sum?
Yes. And so the optimality, as you will see in a second, is based on Fisher information. So I'm ignoring correlations now. In everything I'm telling you in the first part of the talk, I'm ignoring correlations, or I assume that there is an optimal decoder, even though this maximum likelihood is not, who removes the correlation. So
let's say no correlation, I understand. But still, you have a linear relation here between the responses of neurons. You're trying to model the responses now?
The first question is empirical. Does this equation hold? That's the first question. The second question is, assuming it holds, how close those empirically measured weights are to those that one would measure based on the tuning curves and the variability on each cell, assuming, based on the Fisher information basically of each cell, that will lead, assuming an optimal decoder will lead to this. So I should also mention there was, and this is very specialized, so that's why I didn't include in the talk. There was a paper by Alex Pouget about this time that actually said, so the background here is that if you take a network which consists of you know the Wilhelmina paper? Yeah. So let me show you the data and then maybe talk about it afterwards. So the first question was, does this equation hold? So this is two data sets. The blue one is the Chris's data. The other data set came from a previous study. And what I want, first of all, this equation gave very good fits to the data. And this is what happened to the weights as the coherence changed. As you can see, the weights changed. And that's actually opposite from the only computational study that has been done up to date at that time, which is a paper by Wilhelmina and Alex Pouget, that showed that hypothesized ideal neurons actually who just sum the inputs and where the weights are 1 and they don't change with reliability, they will give you optimal multisensory integration. So actually when we first did this analysis, these data deviated from their predictions because their predictions actually said that you don't need to weigh them in a way that these weights change, just sum the population, sum the activity of the cells, and you will get optimal Q integration. Two different data sets. This was done while the animal performed the task. This was done during passive fixation. The result is the same. At the same time, Also, we measure in collaboration with Alex Pouget, we measure, so again, as I told you before, this is not what would have been expected based on the previous theory. So what the difference between this theory and our data is that the neurons are never ideal. The properties of the neurons and how they change with coherence is not what this ideal computation that they had studied assumed. So based on the actual tuning curves of our cells, which means how they change with the stimulus and the variability, we could actually measure the optimal weights based on Fisher information. And this is what the result is. And I want to highlight two properties here. If we took into account the actual properties of our cells, as we did, then unlike this original study by Wei-Jima, we found that the optimal weights actually also are predicted to change with coherence. And we can verify that it's optimal because we can do this decoding, the population decoding, but now, rather than taking the combined responses, now we calculate hypothetical combined responses using those optimal weights, and then, of course, we get optimality out. So we hypothesize, the second thing I want you to notice is that there is a difference between the actual weights we measured and the optimal weights. In fact, our actual weights change more than what predicted by Fisher information. So we hypothesized then that perhaps the reason we saw the suboptimality is because of this difference in the actual empirically measured neural weights from the optimal weights. And so we tested this again by computing hypothetical combined responses using now not the optimal weights like here, but actually using the measured weights. And when we did that, we could reproduce exactly the suboptimality. So I'm almost done with this first part. I just want to go to ask one last question. So again, just to remind you, what I have shown you now in addition to the behavior is that there is this linear equation also describes the properties of the cells, and that those weights are close to optimal but not exactly optimal. And very importantly, they vary with visual motion coherence with the reliability of the signals. So the question is, how does this happen? Because remember, at some point, these multisensory responses are generated by unisensory responses, and this happens trial by trial. So one possibility is that those synaptic weights change, and of course, that will be a very stupid way to do things. So there is another possibility, and the second possibility is based on a network property. And so I would like to briefly summarize the fact that if we use a known widespread nonlinearity, network nonlinearity known as a divisive normalization, we can predict these changes in the weights. And briefly, for those of you who are not familiar with the equation, so we have the two single Q neurons converging onto the multisensory cell. The two weights, as this activity sums, are fixed. They do not change with the reliability. And after there is an exponential nonlinearity here. But the important part is that on each cell, after those two Qs are added with fixed synaptic weights, then this whole population activity is this activity is actually divided by the sum of the population activity, as illustrated here. This is a form of nonlinearity, of network nonlinearity, which has been shown over and over. And just to make a long story short, if we simulate this network, we can actually predict the results about the neural weights. So let me summarize this now. So what I showed you is that, first of all, animals like humans, perceptually, they take into account how reliable the cues are, and they combine them in a near optimal way such that they can maximize the benefit of each of them. I've shown you that we actually followed this property all the way down to the individual cells. And in fact, the population activity of the neurons we've recorded from could predict the behavior. And then finally, I've shown you that we think that the way that the network, this property works is because information about the reliability of the Q is encoded in the population activity. And this is something, probably divisive normalization is an important component of it, and this is work we are currently continuing. So at this point, I would like to shift gears, and I want to talk about the second part. So we've talked about perception. I spent time describing to you the benefit of multisensory processing, but this last part, I'm only going to talk about one sensory signal. The same applies to everything. So we have recorded these neurons while the animal performed the task, but I have not really used this advantage other than compare the population activity with with the actual behavior of the animal. However, because we measure the behavior at the same time, we can ask the question that, in fact, when we look at the activity of the cells, maybe the activity of the cells correlates with the perception. And let me show this to you with an example. This is the response of a cell to the straightforward 0 degrees heading means moving straight forward, which this stimulus contains no information about whether the animal moved to the right or to the left. And as you can see, there is quite a bit of variability in neurons. So if we repeat the same stimulus multiple times, we get all of this wide distribution. However, remember that for a 0 degree heading, the animal makes mistakes, right? Because there is no correct answer. So 50%, typically on average, most animals report 50% right, 50% left. That is, they operate at a chance level. So what happens if we take this activity now and we color code it according not to the stimulus, which is what I showed you up to now, but according to the choice the animal made. So here it's the same data, but now I've used pink for the stimuli for which the animal chose the right target, and cyan for the stimuli that the animal chose the left target. As you can see, there is a difference. So the animal, for the same direction of the stimulus, the animal fired more when the animal made the choice in the cell's preferred direction, which was to the right. This property is quantified using a general C analysis. This is something that has been done for 20 years now, and it is known as choice probability. It has been shown that sensory areas in the cortex, in the visual cortex, and in the somatosensory cortex, they actually have this property. They show weak correlations with a choice. The cells show weak correlations with a choice the animal makes. So this is just one example. I'm going to show you summary data just to make the point that this effect is very small. So 0.5 means no correlation with the choice the animal makes. Many cells show no correlations, all of those white bars. A few cells show significant correlations with the perception that the animal, the choice the animal makes. Overall, the distribution is shifted a little bit to the right. 0.55, it's significant, but it's a very small effect. This is typical of what happens in sensory cortex. This is about the same magnitude of correlations that have been previously shown in area MT, let's say. I showed you this data for the vestibular stimulus conditions just to emphasize. I told you before that there is no primary vestibular cortex. Instead, vestibular signals are found in many areas, and this is an example. MST is a visual area, and nevertheless, you find vestibular signals and you find those correlations with a purely vestibular task.
There is no visual stimulus?
No. This is the vestibular stimulus condition. We get correlations also. I will not show...
No, but there is no visual stimulus.
No, not in this. We have done that also. Most of the data were not collected with a blank screen because there was a fixation target, but we have done this control. We've recorded from a subpopulation of cells in complete darkness. The result is the same. So another property of those choice probabilities is that they actually correlate with the cell's threshold. So here each dot corresponds to a neuron. So the cells which have the higher correlations with the choice, the perception of the animal, are those which have the smallest neuronal thresholds, and that makes sense because these are the neurons which are the most informative for the task. Again, this is a property that has been described before, nothing surprising. Now I'm going to show you what happens if we did this recording in area VIP. As you can see, there are significant, there are much stronger choice probabilities in this case, in this area, although, as you can see, the thresholds are about the same. So I want a little bit to stop here and I want you to appreciate what this means. A correlation of 1 means that if you just listen to the activity of this guy, you can do the task. The animal can just listen to that cell and it correlates perfectly, which means only one cell can actually generate the behavior. Why do we need the rest of the neurons? So we were very surprised to see this. So keep that in mind as we move forward. For now, I want to go back a little bit in time to tell you what has been known about the functional significance of choice probabilities. Yes?
How much before the animals make the choice to the cells fire?
Say it again.
How much before the animal actually behaviorally makes the choice?
The timing of these correlations. You see them throughout the response. For VIP, where you saw the strong correlations, the response even stay high. Those choice probabilities stay high at the end of the stimulus when the animal makes the choice.
But the firing is before the behavior.
We cannot measure timing because our stimulus, this is the problem with the vestibular system. We use a very smooth displacement, so it's not, this task is not to ask this question. But there are other tricks that we can play, and we are working on this now. But you cannot ask timing in this.
Before the animal makes the choice.
Yeah, it happens before the animal makes the choice, absolutely. So the stimulus lasts 1 or 2 seconds. You see it right away. As soon as the firing rate of the cell increases, you see it right away. And it stays, for VIP, it stays up at the end of the stimulus. So let's go a little bit back in history. The first time that these choice probabilities were described was in Bill Newson's lab in the 1990s. They were shown for area MT. And at that time, the hypothesis was that they represent bottom-up processes, meaning that when the task is very difficult and the sensory stimulus is not very informative, then the animal makes mistakes, and it's the activity, it's the trial-by-trial fluctuation in the activity of the sensory cell that actually drives the decision towards the choice that the animal makes. So since then, this idea has been challenged. There is some very good work by Bruce Cumming and also more recently by Marlene Cohen. So there are 2 or 3 labs that have actually asked this question in the visual cortex, and they basically say no, it's a top-down signal, it's a decision or an attention signal that comes back to these areas, and most importantly, you can just see these choice probabilities not because the cell I'm recording from generates the decision, but simply because the cell I'm recording from is correlated with another cell that makes the decision. So this is where we were when we saw these strong choice probabilities in area VIP. But before I go back to area VIP, I want to tell you that the first thing we did actually was to see whether we can see these choice probabilities in early sensory areas. Until up to today, up to the time we did these experiments, all of the reports of significant correlations with the perception of the animal were made in the cortex. So in the visual system, you cannot easily ask this question because if you go to LGN and the retinal ganglion cells, the neurons do not have the same properties. But in the vestibular system, you can, because vestibular signals are very much the same all the way down to the periphery. So we recorded from the vestibular nuclei and the cerebellar nuclei. So just to give you the anatomy, the 8th nerve, which carries the vestibular signals, comes in to the vestibular nucleus in the brainstem, somewhere here, and the cerebellum. And it's these areas that we have recorded from. And I want you to see that we found significant choice probabilities in the brainstem and the deep cerebellar nuclei. I have put them together here because there was no significant difference. This is the vestibular nucleus and the deep cerebellar nuclei. And in fact, if you compare with the cortex, pretty much actually these brainstem and cerebellar nuclei look more like VIP than they look like MST. So strong choice-related activity, which raises the possibility that perhaps that they might represent bottom-up processes if they are found so early in the sensory processing. However, cortical signals project back to these areas, so we cannot make this decision. So right now we are actually recording all the way from the 8th nerve, but to see whether we also see significant choice probabilities. I do not know the answer. So let's go back to area MST and area VIP. So I've told you they are very similar in terms of the sensory properties. They respond to both stimuli. They have similar tuning curves. They have similar neuronal threshold. Really, the big difference is that they show much stronger choice probabilities. So if really these choice probabilities are related to the perception, then we should then expect that if we inactivate these two areas, then we will expect larger deficits when we inactivate VIP than when we inactivate MST. So this is what we did. So this is what I'm going to show you next. We do bilateral injections of either one area or the other area after we mapped it very carefully and we ask the animal to do the task. So what we expect, if an area really contributes to the perception, if we inactivate a big chunk bilaterally, we expect that the animal will become worse. The threshold will be higher when we ask him to do the task after inactivation. So I'm going to show you an example. So this is the behavior of the animal in the visual stimulus condition before inactivation. This is inactivation of area MST. After inactivation, we see a large deficit. It lasts 12 hours later when the animal is tested again the next day, and it has recovered 36 hours later. We found similar deficits, but smaller in the vestibular and the combined, and these are summary data showing clearly that after MST inactivation, we actually see deficits, not just in the visual stimulus condition, but also during the vestibular and the combined. So what happens now when we inactivate area VIP? Remember, they show those strong choice probabilities. Some cells, they fire all the way up to a correlation of 1. As you can see, there is no deficit. In fact, if anything, the only significant effect, it improved, went the wrong direction. So, and this is summary data now comparing the areas. The orange is area VIP, and the blue-green is area MST. So this clearly shows that large choice probabilities are just correlations. They don't mean causality. But can we tell anything else from this data? As I mentioned to you previously when you asked me, we record from as many cells as we can every time we record. Now in monkeys, it's still problematic. We used 2 or 3 single electrodes in the past. Now, after Adam came to the lab, we are using those linear array electrodes that have 16 tips, so we can record from maybe 3 or 4. We try the best we can, but still everything I'm going to show you today are pairwise correlations. So we record often the activity of 2 cells at the same time. This is the tuning curves. This is mean firing rate as a function of the direction of the stimulus. Now we vary the stimulus throughout the horizontal plane. We record from these 2 cells simultaneous. And the correlation between the tuning curves, the mean responses, we define as the signal correlation. And then when we take the trial by trial responses and we normalize the data and then we compare one with the other, we often see significant correlations, and these are known as noise correlations. So this is a case of an example of a pair of neurons with high signal correlation and high noise correlation. So now I'm going to show you summary correlation data. I'm going to show them in the format of noise correlations as a function of signal correlations, because this is what is important. And I want you to notice that in MST, we do have significant dependence, but it's relatively small compared to VIP, where we have a huge dependence, which in many ways parallels the strong choice probabilities versus threshold relationship. So far, what these data suggest is that Bruce Cumming and Marlene Cohen, the people who had said that choice probabilities means nothing, and they just simply reflect noise correlations. Actually, so far the data support this hypothesis, because in an area where we have strong noise correlations, we also have strong choice probabilities. But I do not think that that's actually the whole answer. So very recently, there was a very nice study published by Ralph Hefner and Matthias Berthe from Tübingen. I do not know if you have seen the paper. So what they said, and I think it really provides a new look to the problem, is that it's neither one nor the other. So what they say is that if you have a population of neurons, in our case we have 2, we have area MST and we have area VIP. If you record from these neurons, and you measure correlations, and such that you can measure the covariance matrix, and you know the tuning curves, everything here which is in blue are known quantities that you measure from the data. The tuning curves, the noise correlations, and if you also measure the choice probabilities, which relate the neural activity with behavioral choice, then what you can do is actually go and compute the weights, the decoding weights that you will need from a linear decoder in order to be able to predict the measured choice probabilities given the noise correlations. So I want to pause here a little bit to think about a few things here. If it was optimal decoding, so the issue of noise correlations has become a little bit muffled in the literature, and I know that Jaime has worked quite a bit on this. So for many years, it was thought that noise correlations are bad because they limit information. However, more recently, there has been a study which replicates some of all Jaime's old simulations, showing that that's not necessarily a case, because when the noise is very different from the signal, you can always find an optimal decoder to just get rid of the correlations. So if you compute the optimal decoder here, you can get rid of the correlations. However, you can never predict choice probabilities if you have more than 100 cells. Because if you really have, when your population of cells is high enough, then something must limit the information. Otherwise, you will never see the reflection of the behavior at the level of the cells. So what the Hefner paper basically says is that maybe you have a suboptimal decoder, and by measuring those weights, you can actually see how suboptimal your decoder is. And so let me show you simulations. This is actually work done by a superb graduate student in the lab. He just joined my lab in July, and he gave me these simulations within a month since he joined the lab. So, this is simulations of this network. Our measure MST cells, measure VIP cells. We estimated the covariance matrix, but of course we had to do it based on pairwise correlations. We assumed no interconnections between the two areas, and we know the choice correlations, and this is the result. So this is when you use both MST and VIP. Let's say you get this population threshold based on these weights and linear decoding that we compute. Now, if you inactivate MST, see that the prediction is that you will see a big deficit in your behavior. However, if you inactivate VIP, you do not. So let me give you, because I don't have any more slides, let me give you a little bit the intuition about this. What happens, remember I told you that if these weights are not optimal, that represents a suboptimal decoding. And indeed, the weights we measure to be able to explain these choice probabilities are different from the optimal weights. So the data suggests suboptimal decoding. So the intuition here is that the noise, the way we have measured this covariance matrix, and there are issues with it if anybody wants to discuss this later, what it means is that the noise in MST is not as limiting as the noise in VIP, such that because we have the suboptimal decoding, we really use mostly the MST to do the job and not VIP. So let me now summarize what I have shown you. And again, this is still work in progress. I've shown you that strong choice probabilities do not always reflect causal roles in perception. And I think this is the first time that this has been shown. In the past, the areas that showed significant choice probabilities, actually when like area MT, when Bill Newsom inactivated area MT, he saw deficits in motion discrimination and in disparity discrimination. Second, I showed you that these two cortical areas, even though they look very similar, might have different roles. And I also believe that, in fact, it's extremely important that we measure firing rate variability. And unfortunately, in monkeys, we are still a little bit limited in how well we can do that. And most importantly, the part that actually fascinates me right now is that if really we see these choice signals all the way to the brainstem and cerebellum, no matter whether they are bottom-up or top-down, that means that perception might not just be generated by the cortex, but in fact it might involve loops that go all the way down to early sensory areas. And this is work in progress. And importantly, let me acknowledge the early people who did the original experiments, Yonggu and Aihua. They are independent PIs now. Sheng did the subcortical decodings. Chris did the decoding, and he's now finishing his postdoc. Kashik is the new graduate student, and many of you know this guy. And as I said, Adam, when he comes, he will take, in a way, some of what I told you today to the next step. And because he has done all of this multisensory calibration work. Thank you very much. Yeah.
So your other the cerebellar measurement may suggest that it's simply fluctuations in the sensory input.
It could be bottom up. Yeah, we do not.
Bottom up, very bottom. Simply
so
the sensory organs, so
yeah.
they fluctuate.
So we are recording from the 8th nerve now. And I don't know the answer yet because of a complication. So we went and recorded from the otolith afferents, right? Because this task, it's a linear acceleration task. Unfortunately, we found out later that the brainstem cells that show the correlations with the choice, it's not all the neurons. It's only those neurons that discriminate translation from gravity. Remember, gravity is a form of linear acceleration. It is a linear acceleration. So the otolith organs, they don't distinguish a tilt, whether I'm moving my head like this or whether I'm moving to the right or to the left, because they're both linear accelerations. And this is my cerebellar work. This is related. I have spent quite a bit of time trying to understand how the brain solves this problem. So when we recorded from the vestibular nucleus and the cerebellar nuclei, we found that only the cells that have solved this ambiguity problem show the correlations with the behavior. So it's not a random property. It's really only the relevant cells. And in the literature, that's the idea always has been that the cells that show the stronger correlations with the perception of the animal are those which are the more useful to the perception. You don't want to dilute your perception by having irrelevant cells show this correlation. So the fact that we found that only the cells that have solved this ambiguity problem have this property makes sense. Unfortunately, otolith afferents do not solve this problem because they don't receive. They respond like linear accelerometers. So we spent a year, 2 years recording from the otolith afferents. We did not find choice probabilities, but that does not mean anything because they do not distinguish gravity from translational acceleration. So now we are going back. We are doing now a rotation task because the canal afferents, during rotation, you don't have this ambiguity, and I don't know the answer yet. But it could be, so there are two possibilities. One is that it is really the activity in the organ, and there are ways that you can get correlations in the periphery because each otolith afferent innervates multiple hair cells, and even the hair cells might be correlated with each other because they have the membrane of the fluid and the movement of the fluid and the membrane, which actually might propagate correlations. So it is possible. Alternatively, it is not at the level of the sensor, but it is created centrally when the signal is in the correct format to be useful. But again, then it's like, why does it go all the way back to the brainstem? I mean, attention. Why would it? Yeah. So or perception is not, or it's a loop, right? I was just visiting the Weitzman, and I heard Ahisar talk about the loops. He talks about different loops. He talks about sensory motor loops. But it could be that, in fact, it's the whole brain that participates in the perception. I don't know.
So to discriminate between these options, one could do the following experiment, and maybe you've done some of it, and this is to allow the monkey to prepare for certain manipulation versus not being prepared to them. So you set a context even before the stimulus starts. So you allow a kind of a top-down processes that would affect the sensations. Like what you showed us in the very first slide.
Interesting. So we have not done that, but what Adam has done. So Adam manipulates the perception by he puts a conflict in the stimuli, and then we try to understand how the cues calibrate each other. And so what we saw is that if you put a conflict and if you manipulate the reward, you see shifts in the perception. So he manipulated perception. Then he went to record not just from the cortex. He will show you data from VIP and MST, but he has also recorded in collaboration with somebody else from the brainstem, from these same areas. We see the shifts at the level of the brainstem and the cerebellum. So the receptor has not changed. I mean, I don't know, we didn't record from the receptor. but we see the activity, we see this change in the perception all the way down to the brainstem and cerebellum. Still, it does not separate, though, because it can be top-down signals.
I remember an experiment where they cooled the area V4 and measured in V1. So this is one manipulation.
So this is one thing. So I've thought of doing this, but we don't know how many areas show this. I mean, it could be that it's more than just 2 areas. So it becomes The solution we have chosen to try is to do a manipulation that Bruce Cumming has used to actually start the stimulus, but then manipulate, give a burst of stimulus, and then measure the kernel. Yeah. So this is what we plan to do. Yes. So these cells, these cells we are recording from, they do not respond to eye movements. I should have said that. None of the cells that I showed you in the brainstem and cerebellum, none of them responds to eye movements. There are other cells that respond to eye movements. And there is no other motor response we study because the task is an eye movement task, right? So we have not, I mean, these are correlations, and even how you define the noise correlation. Noise correlation is whatever is not a signal, but what is a signal, you might be wrong in what the signal is, right? So it's a little bit of a difficult thing to answer. So I cannot eliminate that maybe there is something else that is happening, and we see these correlations, but there isn't anything that we could easily identify. We see them in the darkness. We see them. We have even trained an animal to do the task using a motor rather than an eye movement, and we see the same property. So it's not. Yeah. So what are you asking me exactly? Whether this property that perception is, that the correlations with perception are widespread? Yeah, I do not know, because the trick, nobody has shown choice probabilities in cells that do not have the correct properties for the perceptions you are studying. Like in area MT, for example, you must, let's say the animal is trained to do a binocular disparity discrimination task. You won't see the choice probabilities unless a cell is tuned to binocular disparity. So as long as neurons, so my hypothesis right now, and of course I can be completely wrong, is that as long as neurons have the relevant properties, it's not just the cortex. I think it might be a widespread property. I mean, yeah.
It might be a very late question. If the system is so good in overcoming conflict between visual and vestibular system, why do we have seasickness?
Nobody knows. The seasickness is created.
It's still believe that seasickness is because of conflict.
Yes. So seasickness happens when the two cues are in conflict, and some people are more susceptible than in others. So I've always thought, we don't know the answer, but I've always thought that seasickness is related to causality, to causal inference. So there is another, so everything I have presented to you today assumes that these two sensory signals came from the same physical property in the world, right? In this case, I am moving, and because I move, I expect these optic flow stimulus, and I expect this activation of my otolith organs. But in reality, that's not always the case. I can walk, but you can move also, and so the visual stimulus is not always coherent with it. Or you can be in a boat, the boat goes up and down, you are underneath, you don't see. Or for auditory visual, really a real-world example. I showed you previously the situation where you combine the lips and the sound, but you should only do that when there is a high probability that the two cues come from the same source. If I hear Elon's voice, if I see Elon's picture in front of me, but I hear his voice from over there, I might not want to put the two together, right? So this is this theory. A few people recently, not a lot, have actually developed this into an idea that the brain measures a probability of a common cause, and whether you combine the signals is based on that probability. So for me, always the answer is that the brain tries to do the best to solve the problem, to solve this inference problem, but when the answer is that it's two causes, in the case of auditory visual, say, okay, there is one voice and one person, two separate things. But in the case of the visual vestibular, you don't know what solution to come up with. So I've always thought that it's related to this conflict. So the conflict gets too large, you cannot really make the correct inference of what the real stimulus is, and then you get sick. And maybe being sick is a protective way to avoid some of these situations, right? I don't know why it got generated. I don't know why it happens. And it differs from subject to subject. It's not that all of us get sick all the time. Thank you.